Okay, hopefully you can hear me now. I'm Jo Flanagan. I'm the CEO of Women's Health Tasmania, and welcome to our webinar on vaccine hesitancy. So thank you for coming along. I'm still get excited when people from all around Tasmania sign up for these things, and it's very thrilling to have people on King Island and Cape Barren Island and as far away as Geeveston participating with us today. So welcome to you all. This meeting that we're having today is taking place on Aboriginal land across Tasmania, and we want to acknowledge and pay respect to the traditional and original owners of Lutruwita, Tasmania, the Aboriginal community. Through our work here at Women's Health Tas, we work a lot with the elders of the community, the women elders, and they're an extraordinary bunch of women, and we'd like to give our respect to them and to the young folk who are emerging as their new leaders. And finally, we'd also like to acknowledge and extend our respect to any Aboriginal and Torres Strait Islander people who are joining us in the audience today. So everyone has questions about the vaccines and the rollout and how it's all working for us. Our webinar today aims to provide you with information and context so that you both understand it better, but also have the tools to have useful conversations with friends and colleagues and clients. So the presenters will give you an overview and then focus on the common questions that people have, and hopefully they'll be helpful to you in your conversations. They're very good folk. They redid their presentations yesterday based on the questions that we'd received through the registration. So hopefully you'll find some of your questions answered through that. But it is a live Q&A, so if you have questions coming up through the process of the webinar, please use the Zoom Q&A feature at the bottom of the screen. The presenter may answer you in the Q&A chat box, or we might park the questions for the live discussion at the end. But do please ask questions. So now I'd like to introduce our speakers. It's Katie Flanagan and Shannon Melody. I'll start with Dr. Flanagan. She's an infectious diseases physician, and she's a professor at 3 universities, UTAS, RMIT and Monash. She's a world-renowned expert on vaccines, particularly adverse reactions to immunisations. You're going to hear a bit about ATAGI today. That's the acronym for the committee that advises the Australian government on immunisations. And Katie not only sits on that, but she actually chairs the ATAGI committee, which advises the Australian government on COVID-19 vaccinations. Katie and I kindly agreed to have someone who wasn't a Flanagan on the webinar today, and that's Dr. Shannon Melody. Shannon is a public health physician working in the Tasmanian Public Health Emergency Operations Centre. That's the centre coordinating the rollout of the vaccine in Tasmania. So Shannon will speak after Katie. Katie, can I hand over you to take it away?
Sorry, I should unmute. Yes, so thank you very much, Jo. And yes, we're not related at all, you and I. Now, I'm just checking, I'm sharing my screen. And is that looking fine? You can see my slides?
Yes.
Yep, okay, lovely. All right, well, I would also like to acknowledge the Aboriginal people as the original and traditional owners of the land on which we're all meeting, and pay my respects to their elders past, present and emerging. So I'm going to talk a little bit about the Australian COVID vaccine program and hopefully give you some content and some background. And as mentioned, I'm a member of ATAGI, and these are my opinions, not necessarily those of ATAGI. Just to put things into a global context, more than 160 million documented cases now of COVID-19, and almost 3.4 million people are known to have died of COVID-19. There are more than 280 vaccines developed now, which is absolutely incredible, and 100 of those have gone into human clinical trials. So this slide here just illustrates the different approaches that have been taken to COVID vaccine design. And what you can see on the left-hand side is the conventional approaches. So these approaches we've used for other licensed human vaccines, protein subunit vaccines, where you take bits of protein and mix that in with an adjuvant to stimulate an immune response, Below that, we've got virus-like particles. Now, the HPV, human papillomavirus vaccine against cervical cancer, is a virus-like particle. And below that, we've got either attenuated viruses, that's a live attenuated virus, live vaccines such as measles, yellow fever. And below that, inactivated viral vaccines, where you take the whole virus or bits of the virus and inactivate it completely so it can't replicate. But on the other side, we've got the novel approaches. And this is what's been very much exciting about the development of COVID-19 vaccines. At the top, we've got nucleic acid vaccines, either DNA or mRNA vaccines. And below that, viral vector vaccines. These can either be ones that do not replicate at all, they're replication incompetent in the humans, or ones that actually can replicate. And essentially what all of these are trying to do is deliver the genetic code to make the protein of interest in the human cells. Now, I'm going to talk a little bit more about the two vaccines that we have in Australia, rather than go through all the technology and how the other ones work, because I think that's of most relevance right now. So starting with the viral vector vaccines, obviously the AstraZeneca is a viral vector vaccine. And at the top of the figure on the right, you can see you've either got replicating viral vectors, which are injected into the host and can actually replicate inside the cells that they infect, or non-replicating, where they're taken up by what's called an antigen presenting cell. And that's that wiggly one that you can see at the bottom right. And those, they then take over the genetic machinery within inside the cell and lead to production of the protein on the gene that they've carried in. And you can see that both of these are carrying the spike gene. And the spike gene is the gene that encodes for the spike protein. And the spike protein is what actually binds to human cells and allows the virus to enter those cells. So what you're trying to do is get an immune response against spike so that you block the entry of the virus into cells. And that's essentially how it works. They're called neutralizing antibodies, and they're supposed to stop the virus from infecting humans. So the good things about these vaccines is they're very safe and well tolerated on the whole, but we will talk further about some of the safety issues. And they can lead to very high protein expression, often with just a single dose. And so the Janssen vaccine, which is an adenovirus vaccine as well, is just a single dose vaccine. The Oxford AstraZeneca one is actually used a chimpanzee adenovirus, and one of the reasons for that is because we produce antibodies and have natural antibodies to adenoviruses. So they chose one that we wouldn't have antibodies to as their viral vector to carry the gene into the host. But it's actually quite complicated to make these vaccines, and it takes quite a long time to scale up to make them. And every single vector requires slightly different processes, and purification is quite difficult. Contamination can be an issue, so it takes quite a long time to make them. And one of the other issues is, as I mentioned, making antibodies and anti-vector immunity to the actual viral vector, which can actually make the responses less. And secondly, I'm going to talk about the RNA vaccines. So the Pfizer vaccine that we have rolling out is obviously an RNA vaccine. The Moderna one that we have just now made a purchase agreement for in Australia is another example. And as you can see in this figure, they're either non-replicating or self-amplifying mRNAs. And both the Pfizer and Moderna vaccines are non-replicating. Essentially, the RNA, which you can see in blue in the figure, is packaged in a lipid nanoparticle. So it has to be packaged like that because they're very unstable, the RNA, and it needs some way to keep it stable. And those particles get taken up by the cell, and then they go into the cytoplasm and take over the host machinery to make the protein of interest. And then you get an immune response against it. If you have a self-amplifying RNA, it carries a genetic code to allow it to keep reproducing within the cell. So you just get more protein expression with them, but then you tend to get more adverse events as well. They're very easy to design and very rapid to manufacture and scale up. And in fact, if you get the sequence of COVID or even a new sequence of one of the variant strains, you can have a vaccine ready in 4 weeks. The immune response tends to be very robust, but as I said, they can be degraded quite quickly. RNA is very, very unstable, so it needs to be packaged, often requires ultra-low cold temperature storage. And you get quite a lot of an innate immune response stimulated inside that cell to that RNA that's all inside the cell. And we don't know much yet about repeated administration of RNA vaccines in humans because we just don't have the experience. So just to go through the vaccine development pipeline, this is what will have happened with each of the vaccines that are going through clinical trials. They start off with preclinical work, either in vitro or in a test tube, or then going on to animal models, usually then involving a challenge of those animal models to prove that the vaccine protects against being challenged against the virus of interest. You then go into phase 1 trials where they're looking to check that the vaccine appears safe and that it gives an immune response that you're expecting, and that's usually in less than 100 people. And if that is shown to be true, then they ramp up to phase 2, which is normally less than 1,000 people, to get more numbers and more confidence of safety and immunogenicity. And then on to phase 3 trials, where they can test for efficacy as well, to check that this vaccine actually protects against the infection it's intended to. And for COVID vaccines, that's been done in tens of thousands of people, 30, 40, 50,000 people in those trials, which is a very big trial for efficacy studies. Once you've shown that that all works, you can then apply for regulatory approval. And in the case of all the COVID vaccines at the moment, these are provisional approvals. And in Australia, obviously, that's done with the TGA. And then after that, you have to keep watching for safety signals after the vaccine's being rolled out. And in 3 years or so, they can then apply for full approval if everything is looking good and the vaccines seem to be safe and effective and all the phase 3 trials have come through. This summarises, as of the 7th of May, all of the different vaccine platforms that are in either preclinical trials, so the ones that are before you get into the humans, the phase 1, and those in clinical trials. And of those ones in clinical trials, 30 of them have progressed through to the phase 3 efficacy stage, which is absolutely unprecedented in terms of vaccine development, where you're lucky if you get 1 or 2 vaccines developed against a disease. And what you can see is that the protein and peptide subunit approach is the most popular, more than 100 of the candidates using that approach. But all across the board, we've got different candidates coming through using different vaccine approaches. The only one that's not very common is a live attenuated virus. As I said, they take a long time to make and attenuate an actual virus to be sure that it's safe. They're contraindicated in people with immune compromise and pregnant women, but they do elicit very good broad immunity. But I think people are much more interested now in using the new technologies, which are much cleaner and much better probably in the end. And you can also see that the bacterial vectors have not gone into clinical trials either. So this is a bit of a busy slide, but it's just to summarize some of the phase 3 trial results that have come through. And we'd all be fairly familiar with these. So at the top, we've got the 2 RNA vaccines, both showing around 95% efficacy against symptomatic disease 7 days after the second dose of the vaccine. And what was important with the Pfizer vaccine, that efficacy was consistent in those over 65, in people of different ethnicities, and people with stable comorbidities such as diabetes, hypertension, respiratory diseases. Then we've got the 3 adenovirus vaccines that have released phase 3 results. So that's the AstraZeneca one that we have, which showed initially in the very first trials a 70% combined efficacy against symptomatic disease after 2 doses of vaccine. However, what we've subsequently found out is that when you increase the dosing interval, you actually get better efficacy. So when you've got a 12-week dosing interval, it's around 80% efficacious. But importantly, no hospitalizations or severe disease. So 100% protecting you against getting severe disease or coming into hospital. They didn't have very good data at the time in those that were over the age of over 65, but they've now subsequently generated good data on that. The Johnson & Johnson vaccine is another adenovirus, 26, and that's a human adenovirus, 66% efficacy, but a different endpoint here. It was moderate to severe disease 28 days after vaccination, single shot vaccine, but 100% efficacy against hospitalization and death. The Russian vaccine, the Gamaleya Sputnik 5 vaccine, uses 2 adenoviruses, an adeno 26 prime and an adeno 5 boost. And they had 92% efficacy, but there's been some recent publications saying that there's some issues around the potential credibility of those results, and there's some inconsistencies in what they've reported, and they've been changing the data, and they haven't released the actual raw data. So we're not quite sure if this is true that they've got such high efficacy after a single dose, and it seems a little unlikely when you compare it to the other adenoviruses. We then got the Novavax vaccine. And that's a protein vaccine, the spike protein, with a special adjuvant matrix M, and that's around 90% efficacy. And underneath, we've got the Chinese vaccines. One of them is an adenovirus, and two of them are whole inactivated virus. And their efficacy results are all a bit messy. Nothing published yet, but anything between 50 and 80% efficacy with those. But I should warn you, these are interim results. So these trials are not finished yet, and we may find more information as time goes on. And we can't compare the results from different trials because they're in different populations using different endpoints. We don't know how long that protection lasts. And we know that many populations were underrepresented, pregnant women and severely immunosuppressed, for example. And we don't know much about effects on disease transmission, but we're beginning to see that these vaccines do decrease transmission. We know that rare side effects might be missed, and real-world efficacy doesn't always play out the same as what you see in a clinical trial. And this slide demonstrates that. So this is a summary that's just come out of Public Health England, looking at the effectiveness of the two vaccines that we have at 1 and 2 doses against various outcomes. Symptomatic disease, you can see that 2 doses of Pfizer is around 90% efficacy, 1 dose of AstraZeneca up to 70%. However, of course, we don't have data for 2 doses of AstraZeneca because of the 3-month dosing schedule most countries are using. But against hospitalization, mortality and infection, you can see very impressive efficacy. So after 2 doses of Pfizer, we're talking 99 to 99% efficacy. And even after 1 dose of AstraZeneca, really quite impressive efficacy, which is likely to be higher once they've had the 2 doses. And we can see some effects on transmission as well. And what you can also see here is that how confident we are with those data. From a worldwide point of view, 11 vaccines have now got regulatory approval throughout the world across a range of different platforms, and more than 1.3 billion doses have now been given in nearly every country in the world, although without a doubt, the richer countries are taking the greater share of the vaccines. And it is still going to take many years, possibly 5 years, to actually vaccinate the whole of the, or 75% of the world's population. From the COVID program point of view in Australia, the process by which the vaccines were selected was using a SciTag, a Science and Industry Technical Advisory Group, to advise the government rather than the PBAC. And they continue to make those decisions around pre-purchase agreements with companies. And all the clinical and technical advice tends to come from ATAGI, which, as you know, I'm a member of, and the National Centre for Immunisation Research and Surveillance support ATAGI. And the programme implementation on this occasion has been done by the COVID Vaccine Task Force federally in conjunction with the jurisdictions, which is not the way things normally work. And there's those 3 groups, advisory groups within COVID, which are advising the government, and I chair the utilisation and prioritisation group, but there are also distribution, programme implementations and safety evaluation, monitoring and confidence, which is the one that's very relevant to this talk today. As you know, we've in Australia made pre-purchase agreements for a number of vaccines. So for the AstraZeneca vaccine, 53.8 pre-purchase doses. With the Pfizer vaccine, it was only 10 million, but the government have now been able to negotiate 40 million doses. With the Nova vaccine, we have a pre-purchase agreement of 51 million. So that's that spike protein vaccine. But at the moment, that's still undergoing a rolling review with the TGA and has not got provisional approval. So the only 2 with provisional approval are the 2 we're using. Moderna hasn't applied to the TGA yet, but as you would have all heard, another 25 million doses of that have been procured, and the plan to potentially manufacture onshore. And the Johnson & Johnson vaccine is also undergoing rolling review, but there's been no pre-purchase, and we may not buy that one given that it's another adenovirus vaccine. And just finally, that bottom one was the University of Queensland spike protein vaccine, but unfortunately that led to false positive HIV results and had to be abandoned. In Australia, we're using these 2 vaccines. You all know the schedules. It's a different schedule for the 2 because of this need to dose the AstraZeneca vaccine more broadly. And we'll just talk about the thrombosis, thrombocytopenia syndrome, because this is something people are very, very concerned about. So it occurs 4 to 28 days after vaccination and manifests with unusual clots in unusual sites, low platelet counts and bleeding. It's got a very specific characteristics, this condition, and it's completely idiosyncratic. We cannot find any predisposing factors coming up at the moment. So people who have high risk for clotting, PEs, previous pulmonary emboli, previous clots are not necessarily at increased risk of this condition. And if anything, you get lower clotting in people who've had the AstraZeneca vaccine. So what ATAGI had to do was do a very, very careful benefit risk analysis to decide if it was reasonable to continue vaccinating all age groups with the AstraZeneca vaccine. And the issue is that people in younger age groups are more likely to have this syndrome than people in older age groups. And in the end, the risk-benefit equation really suggested that if you're under 50, you should probably not have this vaccine, because at the moment in Australia, the risks would outweigh the benefits. But it is worth pointing out, if we had any suspicion of a wave of COVID coming into the country, the benefits would massively outweigh the risks. And eventually we are opening up the country to COVID. But that was where the recommendation came from. And I thought I'd just put up a few questions here. So are there any no predisposing factors to this? The answer is no, apart from it's more common in younger people. Can you have the vaccine if you've had a previous history of DVT, PE, stroke, immune thrombocytopenic purpura, so low platelets, again from an immunological point of view, or thrombocytopenia, which is low platelets, and you can have it. You can have it if you have risk factors for these. You can have it if you have an autoimmune disease, cancer, immunocompromising conditions. If you've recently had heparin, you can have it. You can have surgery shortly after it and have heparin. And if you've had the condition from the first dose, then you really should not get it again. And in fact, if you have a history of either heparin-induced thrombocytopenia or a central venous sinus thrombosis or splenic vein thromboembolism of unknown cause, then yes, you should not get the vaccine. Although there's actually no evidence to support that, we thought that would be common sense at the moment. Somebody mentioned in their questions about timing with flu vaccine. For all vaccines, we're saying a 2-week gap at the moment. And the only reason for that is lack of data and because we want to be able to attribute adverse events properly. And those recommendations may change in due course. But at the moment, 2 weeks apart from any of these vaccines. Both the vaccines would be considered theoretically safe in pregnancy and breastfeeding, and the animal studies don't support any evidence of harm. But of course now, we're not giving the vaccine, the AstraZeneca, to those under 50, then pregnant women will be getting the Pfizer vaccine. That vaccine cannot integrate into the human genome, cannot cause fetal problems. There's no risk of that at all. So very safe in pregnant and breastfeeding women, and tens of thousands of pregnant women have now received the vaccine. And finally, just should mention the variants. There's no doubt that some of the vaccines don't work as well against the variants, but essentially the main problems are with the South African variant, where the AstraZeneca vaccine is really not as good, nor is the Novavax vaccine, and nor is the Janssen vaccine, but we don't really have much information for the Pfizer vaccine at the moment. So just to summarize, we've got 11 vaccines deployed worldwide, more than 1.3 billion doses have been given. This has never happened in the history of mankind at this scale. All the provisional approvals are based on these interim results. We've got new platforms never licensed before for human vaccines. We've got 2 very safe and effective vaccines and 2 more in the pipeline in Australia. And we always knew first generation ones will not be perfect, but we will also need to protect against emerging variants in the future ones. So I will stop there and go back to here and unshare my slides. And hand over to Shannon, who you've had introduced to you and is going to give you her perspective. Thank you.
Thanks, Katie. I'll just share my slides. Can someone let me know that they can see those?
No, can't see them at the moment, Shannon.
Okay. Sorry for this, just a moment. Okay, can you see those?
Yes, we can see
Yes.
them now, Shannon.
Thank goodness. Good afternoon, everyone. So my name's Dr. Shannon Melody, and as mentioned, I'm currently working within the Tasmanian Vaccination Emergency Operations Centre. So we are overseeing the rollout of the COVID-19 vaccination program in Tasmania. So I'd like to speak in my allocated time just a little bit about the role of the vaccine in the pandemic response, some common questions that we have received, and then I'd really like to delve into vaccine hesitancy because I know that is it's very topical, and hopefully be able to equip you with some tools that might help you have conversations with people around their hesitancy. Okay, so just briefly to frame this all into context, pandemics occur when we have a new novel infectious disease that the human immune system has either not seen before or looks different to what it's seen before. So previously that tends to be influenza because the virus has this ability to shift and drift its antigens, and so the immune system doesn't recognise it. And in this case, we're dealing with a novel coronavirus. The scale of that pandemic depends on a whole host of factors, but might include things like how infectious that pathogen is, how virulent it is, which means how severe the disease that it might cause is. The infectious period, so with COVID-19, we have people who are often infectious in the 48 hours before they become unwell. So that poses a bit of a challenge because it means that people are mixing in community whilst infectious. And then other factors, of course, are the health system and the public health system infrastructure to contain a pandemic. And many of the public health measures that we've used to date in Australia and globally have been very much about minimising spread. So physical distancing, hand hygiene, staying home when unwell, and now more recently, vaccination. However, to really get us out of this pandemic, we need immunity. So we need our population to be immune to COVID-19 or the virus that causes COVID-19. How the vaccine or the role of the vaccine in getting us away from this pandemic or shifting it to a different shifting to a different phase of the pandemic depends on a number of factors. One is coverage, so how much uptake there is of the vaccine. Clearly, if we have a population where we've got 70, 80, 90% coverage, we're in a much better position than where we are right now, which in Tasmania is just under 20% of the population who have had one dose of a COVID vaccine. Related to coverage is how we're able to reach all groups of the population. So that includes pregnant women and children. And at the moment, we're currently not routinely recommending vaccination in pregnant women, and we're not vaccinating under the age of 16. The effectiveness of the vaccine is key, and particularly whether it prevents transmission, whether it prevents mild disease, or whether it prevents severe disease. And we're learning more and more about the COVID-19 vaccines that are currently in use. And as outlined by Katie, we know that these vaccines are very effective in preventing serious disease and death. And there's some very promising data around mild disease and possibly transmission. The other factors that help us understand the role of the vaccine in the pandemic response is the duration of protection that the vaccine gives and its impact on variants of the virus. And we're also considering what the role of a vaccine might be if we were to have an outbreak. So that's work that's currently underway, and of course depends on many factors relating to the nature of the outbreak itself. But I just want to emphasise, and as Katie has stated, it's actually a remarkable feat of science that we're in a position today and this year where we have effective vaccines against COVID-19. So now moving on to some of the frequently asked questions, and these are ones that come to us usually through the public health hotline or in other activities, including at the clinics when people are coming forward to be vaccinated. So Katie's covered off the aspects around, is the vaccine safe in pregnancy and breastfeeding? But the other one that we hear all the time is, how were these vaccines developed so quickly? And linked to that, are they safe? And then, of course, there's also a lot of concern around for people who have a history of blood clots, whether they can in fact safely have the AstraZeneca vaccine. So I'll just add a little bit to what Katie covered around how the vaccines were developed so quickly. a number of things and factors were at play here that meant that we're now in a position just over 1 year on and vaccinating the community against this new virus. So firstly, China was very quick to rapidly sequence the virus itself and share that genetic code with the rest of the scientific world. And so that happened within weeks of the virus first being identified in Wuhan in China back in late 2019. And that's sort of the recipe that scientists were able to quickly use to start developing the vaccines. There's been substantial and unprecedented global investment and priority. And that's really linked to the scale of this pandemic and the impact it's had on health, society, and economy. There's been hundreds of thousands of motivated volunteers that have stepped forward for the trials. There's been unprecedented funding. We've seen overlapping of the phases of clinical trials where phase 1, 2, and 3 would usually occur in series. So phase 1 would occur, then there'd be an assessment about whether to pursue phase 2 and phase 3. Whereas what we saw throughout last year was that those phases were overlapped, which is quite risky from a financial perspective. If it's determined that a phase is unsuccessful, there's financial risk there, but that really points to the global investment and priority that was placed on these vaccines. And also linked to that, pharmaceutical companies started producing vaccines even before phase 3 trials were finished. And finally, unfortunately, because there has been such widespread community transmission, it means that it's been quicker and easier to identify how effective these vaccines are because there's more people who are unwell with COVID-19. So there's some of the factors that have played into how the global scientific community were able to develop these vaccines just so quickly. And to put this in scale, this figure demonstrates some of the infectious agents that we know cause different diseases and the timing between their first discovery and the development of an effective vaccine, which is marked by green. So you'll see that there's some diseases there like malaria where we're still searching for that effective vaccine, despite having known about it since 1880. Right down the bottom, you see COVID-19, and it's really just remarkable, the 1 year turnaround in developing a vaccine after identifying a new pathogen. So that question links into, are these vaccines safe? And as Katie outlined, there's the phases of the trials that then lead to approval, and then we start doing what we call the real world surveillance. And that's something that my team here in the Department of Health are responsible for in Tasmania. So I just wanted to talk about what that looks like in Tasmania so you have a sense as to how we keep an eye on safety in our very own state. So essentially, we have a surveillance system called Adverse Events Following Immunisation Surveillance. And what that means is that if there's any untoward medical occurrence that follows a vaccine, we encourage that it's reported to the Department of Health in Tasmania or to the TGA. An adverse event might not be related to the vaccine itself. It could be purely a coincidence, or it could be very clearly related. It could be an administration error. I'd like to reiterate that vaccines very rarely cause serious adverse reactions, and the vast majority are side effects. So they're the expected side effects that we will feel 1 to 2 days after vaccinations, like a sore arm, a headache, slight muscle aches, and these are things that go away on their own without any intervention. So we have 2 main arms of surveillance in Tasmania. We've got passive surveillance and active surveillance. Passive surveillance requires someone to recognise whether it's a person who's been vaccinated or a healthcare worker, to recognise that there's been an adverse event and report it in. We then collate them, gather further information, and feed that information up to the Therapeutic Goods Administration, who monitors safety on a national scale. We also have active surveillance in place, and this is done through AusVac Safety, run out of NCIS, the National Centre for Immunisation Research and Surveillance in New South Wales. And this is where someone will have a vaccine, and then some days later, day 3, day 8, day 6, week 6, they'll get an SMS which asks them how they feel. That information is then fed up into a national system as well. So this graphic just depicts that we've got members of the public, we've got healthcare providers submitting their adverse events to Tasmania, we share up to the TGA, And the TGA have a whole host of intelligence sources that they use to inform safety. So they get the reports from jurisdictions, they keep an eye on the international scene, they scan the literature, they scan the media, and they pull all this together to detect whether there's any safety signals. And this is exactly what's happening for that rare but serious condition that Katie outlined, TTS. And all of that effort means that we have really great data. So if anyone is interested, there's a weekly TGA safety report. It tells you how many adverse events have been reported. It breaks it down by jurisdiction. And then it does special focuses in on selected adverse events like thrombosis with thrombocytopenia or anaphylaxis, the more serious ones. And also that AUSVAC safety surveillance system I spoke of, which was the SMS that goes out to people and asks them how they're feeling, that means that we have data like this. So it gives us a very clear profile of what people are actually experiencing after their vaccines. So you can see Pfizer vaccine dose 1 on the left and dose 2 on the right. So you can see with Pfizer, and this has been described internationally, after the second dose, people feel a little bit worse for wear and quite commonly experience fatigue, pain at the injection site, and muscle and body aches. For AstraZeneca, we have data for dose 1, but not yet dose 2, and that just relates to the 12-week spacing between doses and that we're not quite at that phase of the rollout yet, but we should start seeing those reports imminently. So you see with AstraZeneca, it's a little bit different. People tend to be more fatigued, headaches, muscle and body aches, and less likely to have injection site pain as compared to Pfizer. But these are all the kind of common side effects that people experience, and it just indicates that their immune system is mounting a response, which is what we wanted to do. So those reports are available if you were to Google AUSVAC safety, and they come out on a weekly basis. The other common question we get asked, and I'll be very brief here because Katie touched on it, but we are frequently, multiple times a day, people are very concerned about their specific medical history and whether that means that AstraZeneca is still safe for them. So people over 50 who have experienced, for example, a DVT, a deep vein thrombosis, or another type of clotting condition, are worried that AstraZeneca may not be appropriate. There are actually just a few scenarios over the age of 50 where we would not recommend AstraZeneca. And that's if someone has a history of a very specific medical condition, the cerebral venous sinus thrombosis, or heparin-induced thrombocytopenia. If someone's acutely unwell on the day of vaccination, we ask them to defer. And that's also because we don't want unwell people mixing within our large vaccination clinics from a infection prevention and control perspective. And of course, if someone's had a history of anaphylaxis to AstraZeneca or the components of the vaccine, then that would be a reason not to have AstraZeneca over 50. So it's actually just a few very select examples of reasons why someone over 50 shouldn't have AstraZeneca. Now to talk about vaccine hesitancy. So firstly, what is vaccine hesitancy? So this is from Professor Julie Leask. She's a leading academic in this field. So she's described it as a motivational state of being conflicted or opposed to vaccination. So it's not related to barriers to accessing vaccine, for example, clinics not being near you or perhaps not being eligible yet. It's actually just a state of considering whether it's right for you. And I just want to emphasize that it's actually completely normal that people have questions about vaccination, especially new vaccines. And so I think that's the first point, is to actually acknowledge that it is normal. And people have vaccine hesitancy on a whole spectrum. So it may be at one end of the spectrum, there's the people that are queuing up around the vaccination clinics ready to get their vaccine. They're the people that would accept all. Then there's some people who accept, but they're unsure. There's people who refuse, but they're unsure. And then there's people who refuse all. I think it's worth noting that there's the term anti-vaxxer that's often thrown around. And I'd like to be clear that that's a very distinct group. That's a group of people who have a very fixed ideology against vaccination generally. And they're quite vocal, and they often have the agenda of sharing that ideology. Whereas hesitancy is actually more common. It's where many of the population sit at the moment. And some recent data that came out suggested in Australia that we've had an uptick in hesitancy in recent months with recent news around AstraZeneca and this rare but serious condition. And it's been cited that about a third of Australians are doubtful about getting vaccinated. So that has real implications for us in terms of responding to this pandemic and eventually opening our borders to the wider world. What we know about people who are more likely to have hesitations around vaccination is that they tend to be women, they tend to be younger, and by younger I mean in their 30s, 40s or 50s. They tend often to not speak English as their first language, or they may have a distrust of government based on previous experience. Noting that not all these things need to line up, but they're just some of the themes that have been observed in some of the hesitancy research. And the reason people are hesitant is really around side effects, so concerns about safety, concerns about efficacy, the sense that there actually isn't a need, and that's really prevalent in Australia at the moment. People look around and they don't see COVID as a threat. So there's really no sense of urgency amongst some. And then people may have concerns about their individual medical or health conditions. So what do you do if you're discussing vaccination with someone and they may be hesitant, or if you're hesitant yourself? I think it's important to acknowledge that everyone assesses risk differently. It's a value-based assessment. And humans use mental shortcuts. So when we're bombarded with complex themes or lots of new information, our brains make shortcuts for us to try and navigate our way through those. So there's a few biases that might occur. If there's a low probability outcome, like TTS, it might be given more weighting as a risk if it's highly publicised. So that's referred to as availability. There's also this human tendency that we would avoid making a decision if we might anticipate that there will be negative emotions attached to it. That's called anticipated regret. There is also this interesting tendency where we would prefer to accept an outcome from doing nothing, i.e. not getting vaccinated, than an outcome from doing something. And that's called omission bias. And we also prefer to avoid taking risks when the outcome is uncertain, and that's called ambiguity aversion. So I think that's just useful to kind of frame human nature and how we handle complex and large pieces of information. So how do you communicate risk at an individual level? Valid consent is absolutely key. So that's about communicating the benefits, the risks, and how that might apply to an individual's circumstances. We need to help people weigh up their individual risks and benefits. And in doing so, it can be helpful to understand the magnitude, but not necessarily push that a risk is acceptable. So it's really useful to explain the magnitude of a risk rather than, well, we just should accept that that's okay. When we're trying to convey risk, we need to try and talk in terms where the risks and the outcomes are similar. And I think this is where we've really struggled with thrombosis with thrombocytopenia syndrome, because you've heard in the media that there's been comparisons to the combined oral contraceptive pill, to smoking, to flying. And to be fair, they're probably not comparable, simply because the outcomes such as a deep vein thrombosis is not the same as TTS. So that's been quite challenging. It can be useful to use visuals to assist with risk and benefit. And here's an example of such visual. So on the left, we've got people as dots. So 1,000 people on the left have not been vaccinated against COVID-19. And on the right, we've got 1,000 people who have had the COVID-19 vaccine. We see that 8 people in those who have been vaccinated will get COVID, and they're depicted as the blue dots. And we see that 90 people who have not been vaccinated will get COVID. You can see that amongst those who have not been vaccinated, 3 may die, whereas none will die in the vaccinated group. 22 will get severe COVID, whereas none will get severe COVID in the vaccinated group. And then there's still some mild disease in the vaccinated group. So that's one way that we can convey risk, is sort of these visualizations. It's also important to note that one of the worst things that we could do is what is called the backfire effect, which is where you bombard someone with your insights, whether they're and they're usually correct. But if you've got someone who's hesitant in front of you and you just bombard them with facts, that often gets people's guards up. So what's recommended is that it's an active listening process where you're trying to understand what their thoughts are around vaccination, why they might be hesitant, and then sharing your knowledge. So this is out of a preprint for the Medical Journal of Australia that will be out very shortly. And it just works through a framework how you could have a conversation with someone who might be hesitant about vaccination. So firstly, they recommend that you start by eliciting their questions and concerns. So you mentioned earlier some concerns about getting the COVID vaccine. Can you tell me more? You acknowledge their concerns. It's understandable that you have some concerns. You set an agenda for the discussion. So let's go through that concern about the clotting risk for you and the possible benefits. How does that sound? Then you share knowledge. So this is where you share what you know about the condition as a risk and the benefits. So the risk from the vaccine is small. It may affect 6 people in a million in your age group. It can be quite serious if not treated early. Here's some information about that rare blood clotting syndrome, alongside with the risk of going to ICU if you acquired COVID-19. I've got some extra information for you to look out for. And then you try and elicit some potential motivation for vaccination. So what might be important to you about having the vaccine now? And then you summarise the conversation, set out some options. So to summarise, your options are not to vaccinate, to wait for Pfizer doses to become available later, or to have the AstraZeneca vaccine now. There's pros and cons of each. Having looked at those, where are you leaning? And then it's about a respectful conversation, but opening the door to further follow-ups. So if there is an outbreak, the risk of COVID is increased, and can we revisit your decision? And then there's all the tools that we have to communicate risk at a public level. So it's about being clear about what we're doing in terms of process and outcomes. So being very transparent about our safety surveillance, using clear messaging, not over reassuring, using credible spokespersons who know what they're talking about, using a diverse platform of communication channels, sustaining trust, using your data to inform action, and keeping an eye on what's working and what is not working in the community. These are some really useful resources. There's a whole host there, and that's because there's a lot, and some of them are specific to specific conditions. So technical advice, the TARGI has their clinical guidance. NCIS has some excellent FAQs, the National Centre for Immunisation Research and Surveillance. Specific questions around allergy, there's ASCIA guidelines, cancer, there's Cancer Australia guidelines. The Australian Government has the Pregnancy and Breastfeeding Decision Aid. There's specific resources for Aboriginal and Torres Strait Islanders. There's some great decision-making support guides. So that visual that I showed you was from Ask, Share, Know. There's also a network called Sharing Knowledge about Immunisation, and then general questions about the rollout itself. The Australian Department of Health, as well as the Tasmanian coronavirus webpage, are good resources. So hopefully that is a useful overview of some tips and tricks around hesitancy. I think useful resources, but having those conversations and being transparent about concerns, but also acknowledging concerns is really key. So I'll hand back to Jo, and I'll stop sharing my screen.
Thanks, Shannon. Just before we move into our general discussion, can I just pick up, you mentioned that people for whom English is not their first language are a group that are experiencing levels of vaccine hesitancy. Are there resources available particularly for groups?
Yes, yeah, there are. So also on the Australian Department of Health webpage, there's all of our materials in the main languages that are known to be spoken in Australia other than English.
Thank you. Katie, I noticed that you were marking some questions to be answered live on the way through.
Yes, I've been busy answering lots of questions there.
So one of them was someone expressing shock at 5 years to 75% of the global population being immunised.
Yes, and by mistake I typed that I would answer that live. I had actually mentioned it, but somehow, anyway, yes, look, no, it's true. And what you've got to think about is the fact that The richer countries are all doing a great job, and 1.3 billion doses have gone into arms, but that's America and UK, and Israel has also obviously done a really good job, and some other countries have done a great job. But we've got all those other countries in the world that are very poor, very difficult health infrastructure. And if it wasn't for the COVAX facility, which is planning to provide 2 billion doses to the poorer nations of the world, I don't think they'd even get a look in. Now, Australia is doing its part in the region. It's starting to provide doses. It is going to have a big surplus of AstraZeneca vaccine, I think, potentially going forward. So, and they did also invest in the COVAX facility, where you put your money in to support also supply to poorer nations of the world. So that is happening, but it's still going to be a very, very huge logistic. And we've also got to think about the children, and when we get into those younger people, and what's happening now in countries where they've rolled out to people over the age of, say, 50 or 60, is that the hospitals fill up with younger people, because you still, as Shannon was explaining, you're still going to get some people that get sick, and there's going to be some people who don't vaccinate, and those people are going to be the susceptible ones. And I think older people understand the risk. They really are at the higher risk, but a lot of younger people think they're fine. Well, we see people die of flu every year. Young people die of flu every year. Even in Tasmania, I deal with several deaths a year, sometimes in 20 and 30-year-olds. It's really tragic. And COVID will be the same, unfortunately.
Mm. And I noticed you'd marked a question about Guillain-Barré syndrome and the link with that.
Yes, so that was somebody's mentioned here about the TGA not recognising it. Look, the TGA are very, very aware of following absolutely every potential adverse event signal in the world. They're working with their counterparts in every country in the world. They've got hotlines with various countries. And the Guillain-Barré thing, of course, we know that certain things that occur naturally will arise among vaccinated people. You've got to see, is there likely to be a link with vaccination? And at the moment, there is no clear link between Guillain-Barré and any of the COVID vaccines. Now, that may change in time. We can't say. There's been a few different conditions where people have worried that maybe there could be a link with the vaccination. And I have meetings with the TGA weekly as part of my role on ATAGI, and I can assure you we are monitoring and watching those all the time. And that's where the TTS signal came up, the thrombosis one, and why the recommendations have been made like they have at the moment.
Okay, thank you. We have a question about if there were a medium to severe adverse effect, is there a body that has been created to support the individuals affected?
I thought Shannon might like to answer that one, actually.
Yeah, so we receive the whole spectrum of adverse events through our surveillance system. If there is a serious adverse event, or one that we call an adverse event of special interest, which is a range of syndromes like Guillain-Barré, for example, or TTS, then there is a mechanism to follow up with that person to firstly gather more information. We need to make sure that as a state, we've got that information as complete as possible so that the information that goes up to the TGA is good quality and can inform their national surveillance. If there's a need for clinical follow-up, it's our duty to make sure that they're linked in. There is currently exploration underway about setting up a specialist immunisation clinic in the state specifically for this purpose, and that's happening in the background. Not sure, Katie, if you want to add to that.
Yeah, look, a couple of things I thought I'd add. So in Australia, we do not have a no-fault compensation scheme for people who've had vaccination. This is something that has been called for by various organisations, and as a member of ATAGI, we've actually asked the government to consider it again. There isn't a huge appetite to do it. And I think if you did have a reason that you would want to get compensation for an injury related to vaccination, you would probably have to go through some civil liability to do that. I don't really understand the legal aspects of it. But what I would say as well is that for all the major adverse events, the TGA will put together a causality panel, a specialist group that will go through the actual signal and will decide how likely they think it is related to vaccination, and will make a determination on that. And I've sat on one of those panels for another vaccine, and I'm going to be on future panels, I believe, for COVID vaccines, and they're very, very carefully considered with a group of experts to make a decision about that. And I think The government's very aware of the fact that there are concerns, and of course with the thrombosis syndrome, we've had a whole host of them in the country, as you would expect, with the AstraZeneca rolling out, and all of those people actually, fortunately, bar one, have been fine and gone home and gone back to fairly normal lives. So that's good. But yes, it is a bit of a gap, and I don't know that it's likely in the coming years that the government will do that. What I do know is they have compensated the drug companies against claims of damage, and the companies were only willing to roll out the vaccines if they did that. So you can't sue AstraZeneca if you have a reaction.
Okay. We had a lot of questions when people registered about how to talk to people, and particularly the people who have very hard views, and but I think, Shannon, your presentation addressed all of that beautifully. But I noted some of the questions that came in weren't so much about us as workers working with individuals, but reflecting on the fact that a lot of the people who registered for this webinar work in respected community organisations, not-for-profits, local government, charities. And some of them were asking, what can our organisation do? What can our public communication, particularly through social media, do to help promote confidence in the vaccines?
I think that's a really good question, Jo, because what we've recognised through our now 12 weeks of experience in rolling out this program is that this very much needs to be a grassroots vaccination program. Community-based organisations are who communities tend to trust. So I think, firstly, just a comment that the role of community sector organisations, charities and such are key in informing local community. In terms of what they can do, I think it's about levering off existing resources and sharing those with your relevant stakeholder groups. I think it's about diversifying. It's a number of things, and that slide that I placed up around communicating with the public, I think, is sort of all the various tools that we should be looking at. So it's about clear, consistent messaging, identifying community champions that the community respect to speak to these issues, using a range of platforms that are accessible to people who speak languages other than English or don't find mainstream services accessible for other reasons. And our public information unit, who head up our coronavirus webpage and the host of materials that come out of that, are very keen to engage. So I'd encourage community sector organisations, if they're wanting to link in, to reach out, because we very much appreciate that as well.
Great, thanks, Shannon. Well, I think
Jo, do you want me to just answer? Sorry, there's a couple here I thought might be just worth covering quickly for people. I've got this one: Canada, Ontario halted giving AstraZeneca to people, and that should be a warning. Well, we also changed our strategy as a result of the risk-benefit analysis, as I explained, and in fact, ATAGI had a meeting, and we have weekly or bi-weekly meetings with the Specialised Immunisation Committee in Canada. So we talked to them when we made our decision that week at ATAGI and asked them why they had halted and what their rationale was. And one of the reasons there is that AstraZeneca was a very minor part of their program, and they were actually relying much more on other vaccines, and so they could make that determination at the time. And so that's why they did it. Our situation was completely different. We were also in a very different epidemiological situation here. So we had a program built up very much around AstraZeneca, and we felt that it wasn't reasonable to give it to people under 50 at the present time. And so that's why we made that decision. And somebody's asked what the schedule is in those over 50. So that is the AstraZeneca vaccine. And there isn't another option at the moment. And many people keep saying, can I ask for the Pfizer if I want it? The answer is no, unfortunately, at the moment. But Pfizer and Moderna likely will be here by the end of the year. But the supply is not guaranteed. Nothing's guaranteed with COVID, and it's not guaranteed that we will not have a wave of COVID sweep through the whole country, a similar situation to Melbourne. That could happen, and that's the risk you run by not getting vaccinated in the meantime. If you wait and get a vaccine of your choice later in the year, that's fine, but yeah, you've just got that risk in the meantime, and it just leaves you in that vulnerable situation.
Okay. Thanks, Katie. I think we've reached the end of our time. We tried to time this so that folk could participate in their lunch hour, so I appreciate that people probably need to go now. We have addressed as many questions as we can. Some of them I was crossing off as Katie and Shannon were talking because the questions were addressed in their slides. Katie and Shannon, we have had a lot of questions. Can people get copies of your presentations?
Yes, that's fine.
That's fine, thank you. We have recorded this session and it will be available on the Women's Health Tasmania website next week. So that would be another way of accessing this information. So thank you very much, Shannon and Katie. Thank you for the work that you do. Thank you for helping us today, but thank you for helping keep our community safe. We really appreciate it. If you'd like to learn more about Women's Health Tasmania, please follow us on Facebook, or you could sign up for our e-news and you'll not only get information about upcoming webinars, but links to former webinars and podcasts and publications. We will be sending out an evaluation for this very, very brief one. It will help us design future webinars to make sure that they meet your needs. So please fill it in. It'll arrive in an email. So thank you, everybody who's tuned in, and thank you very much to our speakers. And goodbye, everyone.
Thank you, Jo. Bye.